700 meters below the surface, a remotely operated vehicle sweeps its lights through open water and finds something that does not look alive at all, just eyes. A faint outline, a distortion in the water where a body should be. The predator is almost completely transparent, and in the deep ocean, transparency is not camouflage, it is a weapon. There is an octopus drifting through the twilight zone right now, with a body so clear that the ocean shows through it. When hunting lights sweep across the water nearby, it changes from transparent to deep red in less than a second. There is a fish with a transparent skull and rotating eyes hidden inside a fluid-filled dome. Scientists studied the species for more than a century without realizing the dome even existed because it collapsed every time the fish was brought to the surface. There is a crustacean that kills another transparent animal, hollows out the body, climbs inside the empty shell, and hunts from within it. And there are transparent colonies drifting through the open ocean that stretch longer than a blue whale, made from thousands of individual organisms functioning as a single predator. None of these creatures are closely related, different body plans, different lineages, different depths, yet all of them arrived at the same solution: disappear completely. Tonight we go into the twilight zone and meet the animals that found a way to vanish inside it. The open ocean begins far above the seafloor and far below the waves, in a space so large and so empty that distance stops feeling meaningful inside it. Water extends outward in every direction without interruption. No reef rises through the dark. No drifting forest softens the water column. No stone interrupts the slow movement of current. An animal entering this space leaves the last solid structure behind and enters a world with no edges at all. Above, faint light filters downward from the surface. Below, darkness thickens into something almost physical. Between them hangs an environment where visibility becomes the defining pressure of life. On land, survival often depends on resembling something nearby. An insect disappears against bark because bark already exists beneath it. A fox vanishes into snow because snow surrounds its body from every side. The open ocean offers no such surface. Water has no pattern to imitate and no texture to disappear against. The background is not blue in the way it appears from a boat or shoreline. Down here, the background is absence itself, a transparent volume through which light travels uninterrupted for enormous distances. In such a place, camouflage changes meaning completely. To remain unseen in open water, an animal cannot merely resemble its environment. It must allow the environment to pass through it. Light entering living tissue behaves with quiet precision. Some wavelengths are absorbed, some are reflected back into the water. Others continue through the body entirely. Reflection creates outlines. Absorption creates shadows. Scattering reveals shape. Nearly all living tissue scatters at least a small amount of light because cells contain boundaries, fluids, proteins, and pigments that bend illumination in slightly different ways. Even the faintest scattering becomes visible in open water, where there is nothing else for the eye to settle on. The transparent animals of the mesopelagic evolved bodies that reduce this scattering almost to nothing. Their tissues approach the optical properties of seawater closely enough that light continues through them with barely any interruption at all. But the ocean does not contain a single kind of darkness. It contains layers, each governed by different forms of visibility. Between roughly 200 and 1,000 meters below the surface lies the twilight zone, where the final remnants of sunlight still descend from above. The light is weak, diffused, and colorless, but it remains strong enough to create silhouettes. Any opaque body drifting through this depth becomes a moving shadow against the dim glow overhead. Predators below have evolved upward-facing eyes, adapted specifically for this geometry of hunting. They do not search for color or detail, they search for interruption. A transparent body removes the interruption entirely. The shadow vanishes, and with it the animal, far deeper, below approximately 1,000 meters. Sunlight disappears completely. The midnight zone belongs to another form of illumination entirely, one produced by living bodies themselves. Tiny flashes pulse through the dark water. Narrow beams sweep slowly through open space. Signals drift upward from organs evolved to create light chemically within flesh. In this environment, transparency becomes more complicated. A bioluminescent beam striking transparent tissue scatters brightly against total darkness, briefly outlining the very body transparency once concealed. The same adaptation that erased silhouettes in the twilight zone can betray an animal completely in deeper water. Different solutions emerge there, darker ones, stranger ones, shaped not by sunlight, but by biological light itself. An estimated 76% of deep-sea animals produce bioluminescence in some form. Communication, mating, defense, navigation, and predation all unfold through pulses of living light beneath the surface. Every transparent body drifting through the mesopelagic exists inside this illuminated arms race. Every adaptation that follows was shaped by it, refined slowly through millions of years in water, where visibility means survival and invisibility means life continuing quietly through the dark. Phronima grips the salp with clawed appendages, built for holding soft flesh in open water. The salp drifts slowly through the mesopelagic, filtering microscopic particles from the current through a transparent barrel of gelatinous tissue, almost blind to what approaches beside it. Phronima strikes without speed or spectacle. It hooks into the body wall, tears through the outer layer, and begins feeding while the salp is still suspended in the water column. Organs disappear first. The inner cavity empties gradually. What remains is not discarded. The crustacean rotates inside the hollow shell, cutting and reshaping the gelatinous tube from within with the same hooked limbs that opened it. The body of Phronima measures only around 2.5 centimeters in length, small enough to drift unnoticed among planktonic life. Yet the structure it creates grows several times larger than itself. The finished barrel, somewhere between 5 and 10 centimeters long, hangs in the water with a strange geometric stillness. Translucent walls, strengthened by secreted chemicals that stiffen the softened salp tissue into something durable enough to survive the movement of open currents. Phronima settles into the hollow chamber and begins using it immediately. Its legs extend from the open ends, paddling rhythmically through the dark water, while the barrel rotates slowly around its body like a transparent capsule carried through the sea. Inside this drifting structure, the female lays her eggs. The young hatch, surrounded by the remains of the animal their mother consumed to build their shelter. Residual tissue lining the barrel becomes their first food. The brood develops while suspended inside a body that no longer belongs to itself, transported continuously through the ocean by the movements of the animal that hollowed it out. Phronima continues hunting during this period, propelling the barrel through immense volumes of empty water while carrying its offspring inside the transparent remains of another living creature. The barrel itself becomes a form of camouflage. Salps are already difficult to detect in the mesopelagic because their gelatinous bodies scatter very little light. Phronima inherits this invisibility by occupying the shell directly. Its own exoskeleton contributes further to the effect. A thin bacterial coating spreads across the surface of the body, functioning almost like a biological anti-reflective film. When bioluminescent searchlights from hunting fish pass across the water column, the coating dampens the scattered return that would normally outline the crustacean against the surrounding dark. The animal does not vanish completely. Nothing in the ocean truly does. But the amount of information returned to the eyes of a predator becomes so faint that the body dissolves into the background before recognition can occur. Its eyes remain the clearest sign that something alive occupies the barrel. Four compound eyes sit embedded within the transparent head, two of them enlarged into enormous forward-facing medial lenses, unlike those of most other crustaceans. The crystalline cones inside these lenses stretch nearly 5 millimeters in length, proportionally vast for an animal so small. They gather dim light drifting through the mesopelagic and direct it inward with remarkable sensitivity. Even these structures reduce their own visibility. Parts of the eyes are partially transparent, formed from glass-like biological material that allows light to pass through while preserving enough optical density for detection. The result is an eye adapted not only for seeing in darkness, but for remaining difficult to see itself. In the late 1970s, the artist H.R. Giger developed the skeletal, elongated form that would become the xenomorph in Alien. Marine biologists and film historians have often pointed quietly toward Phronima when discussing the creature's possible origins. Giger never confirmed the connection directly, yet the resemblance lingers there in the narrow body, the enlarged eyes, the invasive relationship with another living form. Deep water has always carried shapes that resemble imagination before imagination reaches them, and throughout the mesopelagic, countless other transparent hunters continue drifting through the water beside it, solving the same problem through entirely different bodies. In 2004, a remotely operated vehicle drifting through the North Pacific recorded a fish that appeared to carry a bubble of clear water around its head. The animal moved slowly through the dark, small fins, trembling almost invisibly against the surrounding current, while a transparent dome curved over the front half of its body like glass shaped around living tissue. Inside that dome sat two green cylinders pointed upward into the water above. As the vehicle approached, the cylinders rotated forward together, tracking movement beyond the camera lights with an eerie, calm precision. Marine biologists watching the footage realized almost immediately that they were looking at an animal science had misunderstood for more than a century. The fish was Macropina microstoma, the barreleye. Specimens had been collected in nets since the late 19th century, preserved in museums, described in scientific literature, and drawn in anatomical diagrams across generations of marine research. Yet every preserved specimen showed the same strange anatomy, a soft, dark body with two small round structures near the front of the face that researchers assumed were the eyes. The transparent dome visible in the live footage appeared nowhere in the historical record because it could not survive the journey upward through the water column. As the fish ascended toward lower pressure, the fluid chamber ruptured and collapsed before reaching the surface. By the time the animal arrived in human hands, the structure that defined its biology had already vanished completely. The round structures once labeled as eyes were revealed instead to be nostrils. The actual eyes sat enclosed within the transparent dome, tubular and green pigmented, suspended inside a fluid-filled chamber that occupied nearly the entire front half of the skull. For more than 100 years, science had been studying a damaged version of the animal, describing a body altered fundamentally by the act of observing it. The dome itself behaves almost like a biological observatory window. Filled with clear fluid, it gathers and amplifies the faintest available light drifting down from the distant surface far above. Because the tissue surrounding the dome remains transparent, light can enter from nearly every angle, including directly overhead through the fish's own skull. The result is a visual system adapted not for speed or aggression, but for patient detection in an environment where movement appears first as a subtle interruption against darkness. The eyes remain angled upward most of the time. From depths between roughly 600 and 800 meters, the barreleye watches the water column above for silhouettes and flashes produced by living bodies. The green pigment lining the eyes filters away much of the residual surface glow, isolating the colder spectral traces of bioluminescence drifting through the mesopelagic. Prey reveals itself not through shape alone, but through tiny emissions of living light suspended against the surrounding black water. When the fish locates something edible, the movement is slow and deliberate. The tubular eyes rotate forward within the dome, while the body tilts upward from its horizontal position. It rises carefully through the water, aligning itself beneath drifting prey before feeding. Adult barreleyes reach approximately 10 centimeters in length and may survive for around 60 years, unusually long for a fish so small. Their range stretches across the North Pacific, between Japan, the Bering Strait, and Baja California, moving through cold depths where sunlight arrives only as a diluted memory. Researchers suspect the fish feeds among the tentacles of siphonophores, navigating delicately through long curtains of stinging cells to steal paralyzed prey already trapped there. The transparent dome may serve another purpose in this environment, shielding the sensitive eyes from accidental contact with venomous tissue, while preserving visibility in every direction. The barreleye does not vanish through motion or disguise. Its transparency remains fixed, woven permanently into the structure of its head, like part of the ocean itself, a clear chamber suspended quietly between visibility and absence. Japetella diaphana drifts through open water without ever touching the seafloor beneath it. No reef anchors its body, no cave shelters it. The animal spends its entire existence suspended between distant layers of darkness, moving through the mesopelagic and bathypelagic ocean as though gravity itself has loosened its hold. Its body is soft, almost to the point of collapse, built not for force or speed, but for balance. Muscle has been reduced wherever possible. Dense tissue has given way to gelatinous mass, with nearly the same buoyancy as the surrounding water. The octopus neither sinks nor rises quickly. It hangs there quietly, adjusting itself with tiny motions of its arms, while the current carries it through immense volumes of empty sea. Under the dim ambient glow filtering faintly from far above, the body becomes almost impossible to separate from the water around it. Japetella contracts its chromatophores completely, compressing the pigment sacs in its skin until their color disappears from view. The tissue clears. Light passes through the animal with so little interruption that its outline softens into near absence. Only the eyes retain any stable darkness, floating faintly within a body that otherwise transmits the surrounding ocean directly through itself. This transparency works against one kind of hunter perfectly. Predators scanning upward from deeper water search for silhouettes crossing the diluted glow overhead. A solid body interrupts the remaining light descending from the surface and creates a moving shadow visible from below. Japetella avoids creating the shadow at all. Its body transmits the weak light rather than blocking it, leaving almost nothing for upward-facing eyes to detect. But the mesopelagic contains another form of predator entirely, one that does not rely on silhouettes. Fish carrying bioluminescent organs sweep narrow beams through the dark water like living searchlights, probing open space for anything that reflects their light back toward them. Under these directed flashes, transparency becomes dangerous. Clear tissue scatters bioluminescent light brightly against the surrounding darkness, briefly outlining the very body it was meant to conceal. A transparent animal, illuminated directly in black water, can glow more clearly than an opaque one. Japetella solves both problems by changing itself in real time. The instant blue bioluminescent light strikes nearby water. Chromatophores spread open across the skin in less than a second. Transparency disappears. The body darkens into deep opaque red, absorbing blue wavelengths while reflecting almost nothing back outward. To predators using blue light, the octopus ceases to behave like transparent tissue entirely. It becomes optically dark, blending into the surrounding absence rather than scattering illumination through its body wall. Research conducted by Sonka Johnson and Lars Zielinski at Duke University in 2011 revealed how precise this response truly is. Japetella does not react to movement drifting nearby. It does not respond to shadows. Red light leaves it unchanged. Only blue wavelengths matching the spectral output of deep-sea hunting photophores trigger the transformation. The body is tuned specifically to the color of danger most relevant to its environment, ignoring almost everything else moving through the water around it. Measurements taken during these studies showed that transparent Japetella tissue reflects roughly twice as much bioluminescent light as pigmented tissue under direct illumination. The switch to opaque red cuts the reflective signature almost immediately, reducing the optical signal before a predator can resolve the body fully against the surrounding dark. What emerges from this is not simply camouflage, but a biological system where perception and response occur almost simultaneously. Detection of threat becomes inseparable from alteration of appearance. The body senses a specific wavelength and rewrites its own visibility in direct response, shifting between two contradictory forms of concealment, depending on the kind of eye searching for it. And even this remarkable flexibility exists within the limitations of flesh and pigment. Elsewhere in the deep ocean drift animals so optically reduced that adaptation itself becomes unnecessary, their bodies scattering so little light that they approach invisibility without needing to change at all. A remotely operated vehicle moving through deep water will sometimes encounter what appears at first to be a single dark organ suspended alone in the ocean. The camera catches a dim red shape drifting without context, no visible body surrounding it, no fins, no tentacles, only a floating stain of color carried slowly through the current. Then the vehicle changes angle slightly, the lights strike from another direction, and an outline emerges around the red center, faint at first, then suddenly enormous. A body appears where moments earlier there seemed to be nothing at all. These are the glass squids, the family Cranchiidae, distributed through oceans across the planet from depths of roughly 200 meters to more than 4,000. More than 60 recognized species occupy this vast vertical range. Some remain small enough to rest across a fingertip, scarcely 4 centimeters long, even at adulthood. Others grow beyond 3 meters in total length, larger than a human body drifting silently through black water. Despite this variation in size, the family shares the same optical strategy. To within the limits of measurement, they remain almost completely invisible in open ocean. Like octopuses, glass squids possess chromatophores embedded across the skin, pigment sacs capable of changing the appearance of the body. But where Japetella expands and contracts these structures dynamically, glass squids hold theirs permanently collapsed. The pigment never spreads outward across the tissue. The sacs remain compressed so tightly that the skin becomes optically clear, allowing light to pass through with minimal interruption. Muscle, mantle, fins, and internal structures soften into transparency until only a few dense organs retain enough opacity to betray the body's presence. The most visible of these is the digestive gland, a compact, dark red organ suspended near the center of the squid like a small ember drifting inside invisible tissue. The color serves a practical purpose, deeper than camouflage alone. Many prey animals in the mesopelagic produce bioluminescent flashes when disturbed or consumed. If such prey continued glowing inside a transparent predator, the meal itself would illuminate the hunter from within. The red pigmentation of the digestive gland absorbs blue wavelength light, trapping the glow before it escapes outward through the body wall into the surrounding water. Remaining suspended at depth presents another challenge entirely. Constant swimming through deep water consumes energy, and energy is scarce where food drifts infrequently through immense spaces. Glass squids reduce this cost chemically rather than mechanically. Internal chambers within the body fill with ammonium chloride solution, a compound less dense than seawater. The squid achieves buoyancy without muscular effort, hovering almost motionless for long periods while currents carry it slowly through the mesopelagic. Movement becomes minimal. The body drifts rather than swims whenever possible. Even so, transparency leaves one problem unresolved. Eyes absorb light by necessity, making them darker than the surrounding tissue. Viewed from below against faint residual illumination, the eyes cast shadows that can reveal the squid's position. Glass squids compensate with photophores mounted beneath each eye, small biological light organs producing illumination calibrated to match the weak glow filtering through the water above. To a predator looking upward, the shadow softens and dissolves into the background brightness surrounding it. The system is imperfect in a strangely revealing way. Research conducted at the University of Pennsylvania found that the photophores scatter light inefficiently, producing leakage and optical noise that should theoretically compromise the camouflage. The emitted glow does not behave with clean precision. It bleeds unevenly into surrounding tissue. Yet the adaptation functions well enough to persist across evolutionary time, regardless. The ocean does not require elegance from these bodies. It requires survival, and survival tolerates approximation where perfection would cost too much. So the glass squid continues drifting through darkness, with most of its body surrendered almost entirely to transparency, concealing the few remaining visible structures with chemistry, pigmentation, and light generated from within itself. Around it, other transparent lives move through the same water in forms even stranger, where invisibility no longer belongs to a single body at all, but to colonies stretched across distances longer than many whales. A siphonophore begins as many bodies instead of one. Thousands of individual organisms, each genetically identical, attach themselves into a drifting chain where every member performs only a single task. One zooid pulses rhythmically to move water and generate propulsion. Another carries reproductive cells. Another digests captured food. Others extend long, stinging tentacles into the surrounding current. None can survive independently for long. Together they become something that behaves as a single animal, despite containing no single body that could truly be called the whole. The colony coordinates itself without a brain. No central organ oversees the movement of the chain. Information passes through electrical impulses and chemical gradients traveling between connected zooids, spreading instructions slowly along the transparent body like signals through living glass. Smaller swimming zooids clustered near the front influence direction, while larger structures farther back generate forward momentum. Digestion unfolds simultaneously with movement. Reproduction occurs while prey capture continues elsewhere along the colony. The siphonophore performs all the functions associated with animal life at once, distributed across thousands of specialized bodies suspended together in open water. Most siphonophores remain nearly invisible in their environment. Their tissues are gelatinous and water-rich, scattering very little light as they drift through the mesopelagic. Seen from even a short distance away, the colony dissolves into the surrounding ocean almost completely. Only the faintest distortions betray its shape, long translucent strands hanging through darkness with no obvious beginning or end. Prey moving nearby encounters the structure not through sight, but through contact. Trailing behind the colony are curtains of fishing tentacles lined with nematocysts, microscopic harpoon-like cells held under immense internal pressure. When triggered by touch, these structures fire in roughly 700 nanoseconds, among the fastest cellular actions known in biology. Paralysis follows almost immediately. The siphonophore does not pursue prey actively through the water. It occupies a volume of ocean and waits while currents carry smaller animals into the nearly invisible field of tentacles suspended around it. Contact occurs first. Recognition never arrives at all. On the 16th of March in 2020, researchers aboard the Ningaloo Canyons expedition operating off the coast of Western Australia encountered a siphonophore unlike any previously recorded. The colony belonged to the genus Apolemia and had coiled itself into a vast spiral feeding posture drifting through the dark water. The outer ring alone measured approximately 47 meters across. When researchers estimated the full uncoiled length of the colony, the figure reached as much as 119 meters, making it the longest living organism ever documented. Most siphonophores remain far smaller, typically between 20 centimeters and 1 meter in total length. The enormous Apolemia colony was unusual but not biologically impossible, because siphonophores possess no fixed upper limit to growth in the way many animals do. As long as conditions remain favorable and sections of the colony continue regenerating successfully, new zooids can continue forming and extending the structure farther into surrounding water. The idea of death becomes difficult to apply cleanly to such an organism. If large sections of the colony are destroyed by predators or turbulence, remaining zooids continue functioning. New sections regenerate gradually. Individual zooids die continuously as part of the colony's normal existence, replaced by others generated through ongoing growth. The siphonophore persists not through permanence of its parts, but through continuity of the collective structure itself. To drift into one unknowingly means entering a body too diffuse to perceive. A fish larva or crustacean moves through apparently empty water until a single transparent thread brushes against its surface. The nematocysts fire. Paralysis spreads through the prey before any larger structure becomes visible around it. The colony gathers the immobilized body inward through currents and coordinated contractions, feeding quietly while the surrounding ocean remains visually unchanged. The siphonophore achieves invisibility partly because so little of it exists in concentrated form at all, its body dispersed thinly across enormous distances of water. Elsewhere in the mesopelagic, other transparent lives rely on something even subtler, concealment built not from scale or dispersion, but from the microscopic structure of the tissues themselves. Far below the waves, where the final residue of daylight filters downward through hundreds of meters of water, the ocean never becomes fully dark. A dim overhead glow remains suspended above the mesopelagic, like distant weather seen through thick glass. Predators living beneath this layer evolved eyes directed permanently upward into that faint brightness, searching not for color or texture, but for interruption. Any body crossing between the hunter and the surface creates a silhouette, sometimes only a slight darkening against the diluted light overhead, yet enough for detection. Even transparent tissue fails completely to erase this problem. No living body transmits light perfectly. A trace of shadow always remains. Some animals solve this not by disappearing further, but by generating light themselves. Rows of tiny photophores line the undersides of mesopelagic fish and squid, organs capable of producing controlled bioluminescent glow across the belly surface. The light emitted points downward into the darkness below, while the animal drifts between predator and surface glow. If calibrated correctly, the brightness matches the faint illumination descending from above closely enough that the body's silhouette dissolves into the background entirely. The hunter below no longer sees interruption, it sees continuity, light blending seamlessly into light. The precision required for this is extraordinary. Brightness alone is insufficient. The wavelength of emitted light must correspond to the filtered blue frequencies penetrating through seawater from the surface. The angle of emission matters as well, because light disperses differently depending on viewing position. An animal rising even slightly upward through the water column enters brighter ambient conditions and must adjust its own photophores immediately to compensate. Descending again requires dimming. The calibration unfolds continuously as depth, current, cloud cover, and time of day alter the faint light field surrounding the body. Among the most successful practitioners of this strategy are the lanternfish. More than 250 species distributed across every major ocean on Earth. Most remain small enough to fit comfortably in a human hand, silver-bodied and narrow-eyed, carrying ordered rows of photophores along the belly like constellations embedded beneath the skin. By biomass, lanternfish may be the most abundant vertebrates in the oceans, existing in numbers so immense that they shape entire marine food webs while remaining almost unknown outside scientific circles. In the early 20th century, naval sonar operators encountered something strange moving through deep water beneath their vessels. Instruments repeatedly detected what appeared to be a false seafloor suspended hundreds of meters above the actual bottom. Stranger still, this phantom layer rose toward the surface each night and descended again at dawn. For years, the phenomenon remained unexplained. Eventually, researchers discovered the moving boundary was biological, composed largely of lanternfish, accompanied by transparent crustaceans, gelatinous drifters, and drifting cephalopods migrating vertically through the mesopelagic in immense synchronized numbers. The deep scattering layer, as it became known, represented the largest daily animal migration on Earth. Billions of living bodies, rising and falling through the ocean together, beneath the notice of almost everyone on the surface. Hatchetfish refine counter-illumination with remarkable sensitivity. Their bodies are compressed into thin metallic shapes viewed most often from below, where upward hunting predators search for shadows against the distant glow overhead. Photophores lining the belly adjust continuously as the fish changes depth, altering intensity in real time to preserve a near-perfect optical match with surrounding light conditions. The fish does not simply emit light. It measures the environment constantly and rewrites its own visibility moment by moment. The process resembles active noise cancellation more than traditional camouflage. Instead of concealing a signal beneath texture or color, the animal produces a counter-signal precisely calibrated to erase the visual trace of its body. The strategy depends entirely on accuracy. If the photophores glow too brightly, the fish appears outlined against darkness. If the wavelength shifts incorrectly, the emitted light separates visibly from the ambient field around it. A failed match draws more attention than silence ever would. And still, the system persists across hundreds of species through millions of years of refinement. Bodies learning to erase themselves with light while drifting through water, already filled with living illumination, moving silently in every direction. The deep ocean did not become illuminated all at once. Light appeared gradually through millions of separate evolutionary decisions, each one altering the environment for everything nearby. A predator capable of generating a narrow bioluminescent beam gained an advantage in darkness because hidden prey could suddenly be revealed against black water. Transparent bodies, drifting safely beneath faint ambient glow, became newly vulnerable the moment directed light entered the system. Tissue that once erased silhouettes now scattered illumination outward like suspended glass. The response emerged slowly across countless unrelated animals. Some refined transparency further, reducing optical scatter until barely any reflected light escaped the body at all. Others developed counter-illumination, producing their own glow along the belly surface to erase the shadows cast against the dim water overhead. Japetella evolved the ability to abandon transparency entirely under direct blue illumination, transforming its body into deep absorbing red before the reflected signal could betray it. Phronima layered its exoskeleton with anti-reflective bacterial films that dampened the return from passing searchlights. At still greater depths, where directed bioluminescent beams became more important than residual surface glow, some species abandoned transparency almost completely and evolved ultra-black skin capable of absorbing up to 99.5% of incoming light. Every adaptation altered the selective pressure surrounding it. Every solution generated a new problem for something else living nearby. An estimated 76% of mesopelagic species produce bioluminescent light in some form. Communication pulses through the water in coded flashes. Courtship signals shimmer briefly between drifting bodies. Defensive bursts erupt when predators strike. Hunting beams sweep across darkness, searching for reflected movement. In this environment, bioluminescence is not a rare curiosity, but one of the dominant ecological forces shaping the entire oceanic midwater ecosystem. The arms race is not occurring inside the environment. It is the environment. The chemistry underlying this light remains elegant in its simplicity. Within specialized organs, a compound called luciferin reacts with oxygen in the presence of an enzyme known as luciferase. The reaction releases photons directly from living tissue without producing significant heat. Animals regulate the process by controlling oxygen flow into the reaction chamber, switching the light on or off almost instantly, altering brightness, pulse duration, and direction with extraordinary precision. Some species emit diffuse clouds into the surrounding water. Others focus light through lenses and reflective structures into narrow beams resembling underwater flashlights. Almost all of this illumination appears blue. Seawater absorbs longer wavelengths rapidly, especially red light, which disappears within only a few meters of travel. Blue wavelengths penetrate farthest through the ocean, sometimes reaching hundreds of meters before scattering away completely. Evolution repeatedly converged on blue bioluminescence because blue simply travels farther in deep water than any other visible color. The bodies adapted to survive in this environment became tuned to the same spectrum. Japetella responds specifically to blue illumination because blue dominates nearly every meaningful predatory signal in the mesopelagic. Red light carries little ecological significance there. For most animals at depth, it barely exists at all. One predator exploits this limitation directly. Malacosteus niger, the stoplight loosejaw, produces red bioluminescent light invisible to almost every nearby species. Its body contains specialized visual pigments capable of detecting the same red wavelengths it emits, allowing the fish to illuminate prey without revealing the beam itself. Small crustaceans and fish drifting nearby remain unaware they have entered another animal's field of view because their eyes cannot register the color surrounding them. The loose jaw hunts inside a private channel of illumination accessible almost exclusively to itself, broadcasting on a frequency hidden from nearly everything else in the ocean. Across millions of years, the cycle has continued uninterrupted. Light creates visibility. Visibility selects for concealment. Concealment drives more sophisticated illumination. Countermeasures emerge in response, followed by refinements layered atop earlier refinements until entire bodies become records of ancient optical conflicts carried forward through inheritance rather than choice. The transparent creatures drifting through the mesopelagic today are not final solutions to this process. They are temporary outcomes inside an evolutionary contest still unfolding silently through the water around them, shaped by forces far older than the animals currently carrying them. Long before fish moved through the oceans, before vertebrate eyes evolved to scan open water for silhouettes and flashes of living light, comb jellies were already drifting through ancient seas in transparent bodies. Fossil evidence places the ctenophores more than 500 million years in the past, among the oldest surviving animal lineages on Earth. Their form emerged so early in evolutionary history that some researchers suspect they may predate most other complex animal body plans entirely. For hundreds of millions of years, they have hunted in nearly invisible bodies, while the rest of marine life changed around them. Despite the resemblance, comb jellies are not jellyfish. They belong to an entirely separate phylum with a different evolutionary origin, different body architecture, and no stinging cells at all. The gelatinous transparency shared between the two groups emerged independently, two unrelated lineages arriving at similar solutions under the same physical pressures of open water. A comb jelly carries no venom. It does not pulse through the sea with the contractile bell movements of a true jellyfish. Its biology follows another path entirely, quieter and stranger. Most of the body consists simply of water. Approximately 95% of a comb jelly is fluid held inside an almost impossibly delicate arrangement of cells and gelatinous tissue. Internal structures remain minimal, dense organs nearly absent. Light entering the body encounters so little interruption that the animal approaches optical invisibility more closely than almost anything else in the ocean. In calm water under weak illumination, a comb jelly can disappear so completely that only movement reveals it at all. Running along the body are 8 rows of microscopic cilia called comb rows, thousands of tiny hair-like structures beating rhythmically in coordinated waves that propel the animal through the water without fins or jets. When light strikes these rows at certain angles, the cilia diffract it into shifting spectral colors. Rainbow bands ripple slowly across the body, not produced chemically through bioluminescence, but generated physically through the same optical principles that create color on soap bubbles or peacock feathers. In darkness between illumination events, the colors vanish instantly, and the body returns to near absence. Every comb jelly is a predator. Some species extend 2 long retractable tentacles into surrounding water, each lined with colloblasts, adhesive cells that burst open on contact and glue prey into place. Tiny crustaceans and drifting zooplankton become trapped against the sticky surface, while the tentacles reel inward toward the mouth. Other species abandon long tentacles entirely. Lobate comb jellies such as Mnemiopsis expand broad oral lobes from the front of the body, sweeping prey inward continuously as they drift through dense concentrations of plankton. Then there is barreleye, a comb jelly with no tentacles at all, only a vast, expandable mouth capable of engulfing other comb jellies whole. Transparent predators. Swallowing transparent predators inside bodies composed almost entirely of seawater. In the 1980s, Menemyopsis leidy entered the Black Sea accidentally through ballast water discharged from cargo ships. Without natural predators present, the comb jelly population expanded explosively. Zooplankton disappeared under relentless feeding pressure. Fish larvae lost their food source almost entirely. Native fish populations collapsed across large portions of the sea, while commercial fisheries deteriorated rapidly. The ecological disruption spread through the food web with astonishing speed, driven largely by gelatinous predators that many people would never have recognized drifting beneath the surface. The imbalance eased only after another comb jelly arrived. Barreleye ovata, itself a predator specialized for feeding on other comb jellies, began consuming Menemyopsis in sufficient numbers to reduce the outbreak gradually over time. One transparent lineage brought the system apart, another helped stabilize it again. Comb jellies range from forms scarcely larger than a millimeter to drifting bodies approaching 1.5 meters in length. Across all of them, the underlying strategy remains ancient and remarkably consistent. They survive by becoming mostly water, reducing themselves optically until the boundary between animal and ocean nearly disappears altogether. It is the same adaptation that has allowed them to persist through geological ages, and the same adaptation that makes them so fragile and difficult to study once removed from the sea that shaped them. A transparent body can transmit light through skin, muscle, gelatinous tissue, even sections of internal anatomy. But the eye remains a permanent contradiction inside the design. Vision depends on absorption. Photons entering an eye must be captured by light-sensitive cells and converted into neural signals. The moment light is absorbed instead of transmitted, visibility appears. An eye cannot function without becoming optically dense. In the mesopelagic, where so many animals evolved toward invisibility, the eye persists as the one structure that refuses to disappear completely. Different species solve this problem in remarkably different ways, each revealing a separate compromise between sight and concealment. The glass squid surrounds the problem with light. Beneath each eye sits a photophore calibrated to emit illumination, matching the faint ambient glow filtering downward through the water column. Viewed from below, the dark, circular shadow of the eye softens into the surrounding brightness generated around it. The eye itself remains fully present, absorbing light continuously inside the transparent body. Yet, the optical signature it would normally cast outward is reduced by the carefully balanced glow beneath it. The camouflage does not remove the structure. It cancels the contrast surrounding it. The barreleye approaches the problem architecturally. Instead of concealing the eyes directly, the fish encloses them inside a transparent, fluid-filled dome, integrated into the shape of the head itself. The tubular green eyes remain visible inside the chamber, rotating quietly beneath the clear surface, but the dome transforms them into part of a larger optical system rather than isolated dark organs exposed against open water. Visibility becomes distributed across the entire structure. The eyes no longer appear as interruptions inside transparency. They become suspended components within it. Phronima follows another path entirely. Its enormous compound eyes are built partly from transparent, crystalline material that transmits portions of incoming light instead of absorbing everything that strikes the surface. The elongated cones inside the eye, some extending nearly 5 millimeters in length, retain enough optical density for low light detection while reducing the amount of reflected scatter escaping back into the surrounding ocean. The result is not a fully invisible eye because no such thing exists, but an eye whose presence leaks outward far less aggressively than conventional tissue would allow. The leptocephalus larva resolves the problem developmentally rather than structurally. In its earliest transparent stages, the body remains so small and gelatinous that the eyes occupy only a minor fraction of the total anatomy. Their optical density exists, but at scales where the rest of the body contributes almost no additional visibility. As the larva matures, blood develops, muscle thickens, and transparency fades gradually. By adulthood, the eel becomes fully visible compared with the drifting larval stage, and the eyes cease to represent the dominant optical problem because the body itself has already surrendered invisibility entirely. Japetella bypasses the issue temporarily by abandoning transparency during moments of danger. When blue bioluminescent illumination strikes nearby water, chromatophores expand rapidly across the skin and the entire body darkens into opaque red. The eyes disappear into the larger field of pigmentation surrounding them. Their individual visibility no longer matters because the camouflage strategy has shifted away from transparency altogether and toward absorption. What emerges from these solutions is a deeper pattern running through nearly every transparent species in the mesopelagic. Perfect invisibility does not exist. Somewhere within every transparent body remains at least one unresolved structure: an eye, a digestive organ, a cluster of bioluminescent prey glowing faintly through tissue. Something dense enough to interrupt the optical illusion. Evolution does not search for perfection. It manages compromise. Each species negotiates differently with the one part of itself it cannot fully erase. The ocean fills with bodies built around these negotiations, each adaptation solving one problem while exposing another somewhere deeper inside the anatomy. And even when the eyes are hidden successfully, another difficulty waits within the transparent body itself, because many prey animals continue glowing long after they have been swallowed. Somewhere in the mesopelagic darkness, a transparent predator swallows a living animal that still carries light inside its body. The prey dies, digestion begins, yet the glow does not stop immediately. Blue pulses continue flickering from within the stomach, trapped now behind tissue that was evolved specifically to transmit light rather than block it. For a brief period, the predator becomes visible from the inside outward, a drifting lantern suspended in otherwise black water, announcing its position to every larger hunter nearby. This vulnerability shaped the anatomy of transparent animals as deeply as transparency itself. The open ocean does not permit a single solution to remain uncomplicated for long. Every adaptation creates another problem inside the body that carries it. The glass squid reveals this compromise clearly. Most of its form remains almost perfectly translucent, the musculature pale and diffuse, the outer tissues transmitting surrounding light with so little interruption that only fragments of the body appear visible at certain angles. Yet near the center hangs a dark red digestive gland, dense and unmistakable against the otherwise absent anatomy. The color is not decorative. In the deep ocean, red functions almost like black because blue wavelengths dominate mesopelagic bioluminescence, and red pigment absorbs them efficiently. When the squid consumes glowing prey, the digestive gland acts as a biological curtain wrapped around the stomach contents. The meal continues emitting light for a time, but the glow is absorbed before it escapes through the transparent body wall. A similar compromise appears inside Bathycirofosteri, a deep-sea comb jelly drifting through water that rarely receives sunlight. Its gelatinous body approaches optical invisibility so closely that under weak illumination, the animal can seem less like a structure and more like a distortion in the surrounding current. Only the stomach interrupts the illusion. There, deep red pigmentation lines the digestive region, specifically where bioluminescent material accumulates after feeding. The rest of the animal remains transparent. Only the place carrying dangerous light becomes darkened. Several mesopelagic fish species solve the same problem with even greater contrast. Their outer tissues remain pale and translucent, while the stomach lining itself develops heavy pigmentation dense enough to contain internal luminescence entirely. Clear skin surrounds a black internal chamber. From outside, the fish remains difficult to detect against open water. Inside, swallowed light disappears into a sealed compartment designed to prevent the body from betraying itself. The leptocephalus larva may avoid the problem altogether through diet. These drifting eel larvae spend months, or even years, suspended within ocean currents, while maintaining bodies so transparent that the faintest internal glow would expose them immediately. The leading explanation for their feeding behavior suggests they consume marine snow, microscopic fragments of decaying organic matter descending slowly through the water column. Marine snow does not produce bioluminescence. A transparent larva feeding this way remains dark internally, preserving the invisibility its fragile anatomy depends upon. What emerges from these different strategies is a pattern of continuous correction. Transparency solved the silhouette problem created by open water. Transparency then exposed predators to the danger of glowing prey inside their own stomachs. Dark digestive tissues evolved to compensate for the weakness introduced by invisibility itself. Every refinement produces another vulnerability waiting quietly beneath it. The process resembles an endless internal negotiation between concealment and survival. A body becomes transparent and must darken the gut. Eyes remain visible and require counter-illumination or structural masking. Bioluminescent searchlights appear in the water, and transparent animals respond with pigments, coatings, or behavioral shifts. No adaptation exists alone. Every structure inside these animals carries the memory of another problem that came before it. And beneath all of those adjustments remains a deeper mystery still, because even the darkest stomach lining depends on a far more delicate achievement. The transparent tissue surrounding it must first be built at the cellular level with extraordinary precision, matched so closely to seawater that light passes through living flesh almost as though no boundary exists there at all. The difference between a visible body and an invisible one can exist at a scale smaller than a cell. Deep in the mesopelagic, transparency is not produced by magic, nor by emptiness, but by extraordinary control over the way light moves through living tissue. Every transparent animal drifting through open water carries an anatomy engineered around one central requirement: reduce scattering until the body and the surrounding ocean become almost optically identical. Light scatters whenever it crosses boundaries between materials with different refractive indices, the optical property describing how strongly a substance bends incoming light. Skin, muscle, blood, fat, cartilage all bend light differently. Each transition creates tiny reflections and distortions. Enough of these interruptions together produce visibility. The engineering challenge for transparent life is therefore not simply becoming clear, but eliminating as many optical boundaries as possible from the body itself. Gelatinous animals solve this by approaching the composition of seawater directly. Comb jellies, siphonophores, salps, and several deep-sea jellyfish contain bodies built largely from mesoglea, a water-rich gelatinous material whose refractive index differs from seawater by only 1 to 2%. Light entering these tissues encounters almost no meaningful transition between ocean and body. Instead of bouncing outward, it continues through as though the animal were merely another region of water with slightly altered density. In weak illumination, the boundary separating organism from environment nearly disappears. Glass squids achieve the same outcome through another route. Like octopuses, they possess chromatophores across the skin, pigment sacs, capable of changing the body's optical appearance. Yet, unlike octopuses, glass squids keep these chromatophores permanently collapsed. Pigmentation vanishes from the outer surface almost entirely, removing one of the strongest scattering layers normally present in cephalopod tissue. Beneath the skin, the body remains heavily water-based, its internal refractive properties approaching those of the surrounding sea closely enough that most incoming light passes through uninterrupted. The leptocephalus larva simplifies the body even further. Muscle remains sparse and underdeveloped during this drifting stage of life, replaced largely by transparent gelatinous filler. More importantly, the larva lacks red blood cells. Hemoglobin, the oxygen-carrying molecule responsible for the color of vertebrate blood, is optically dense and highly disruptive to transparency. Without it, the tissues lose one of their strongest sources of visible absorption. The larva drifts through open water, carrying almost no structures, dense enough to interrupt incoming light significantly. Inside the barreleye fish, the transparent dome surrounding the eyes depends on another form of optical matching. The chamber is filled with fluid whose spectral properties closely resemble the surrounding seawater. Instead of scattering available light, the dome guides and amplifies it, functioning almost like a biological lens integrated directly into the skull. Light entering from above continues through the chamber smoothly enough that the structure remains difficult to distinguish from the water surrounding it. Phronima refines transparency at the level of the eye itself. Its crystalline visual cones possess refractive profiles shaped to transmit wavelengths the eye is not actively using, while still preserving sensitivity to dim mesopelagic illumination. The result is a visual organ that gathers information while reducing the optical signature normally associated with light absorption. The eye remains visible in absolute terms, but far less visible than conventional biological tissue would permit. Across many transparent deep-sea organisms, refractive indices remain within 1 to 2% of seawater itself. At this degree of matching, less than 0.1% of incoming light scatters during passage through the body. The difference falls beneath the detection threshold of many deep-sea visual systems, even those evolved specifically to locate prey in darkness. The precision involved is measurable now through optical analysis and deep-sea observation. But for most of scientific history, these structures remained almost impossible to study properly. Transparent animals collapse, rupture, deform, or dissolve during ascent toward the surface. Their invisibility depends partly on remaining alive within the pressure and chemistry of their native environment. Science learned how these bodies work only after cameras and instruments finally descended into the depths where the animals already existed, still drifting invisibly through water that had concealed them for millions of years. For most of scientific history, transparent deep-sea animals became less understandable the moment they were caught. Their bodies depend on pressure, darkness, stable temperature, and the chemical balance of the water surrounding them. Remove them from that environment, and the structures that made them extraordinary begin collapsing almost immediately. Flesh tears, chambers rupture, gelatinous tissues liquefy into residue. Optical properties disappear before the animal even reaches the surface. The barreleye fish revealed this problem more clearly than almost any other species. For over a century, specimens collected in deep trawl nets appeared to possess two small rounded structures near the front of the face, interpreted by researchers as the eyes themselves. The transparent dome enclosing the actual eyes never appeared in preserved samples because the chamber collapsed during ascent. Pressure decreased, the fluid cavity ruptured, and the dome vanished before scientists could examine the fish intact. What remained was a damaged anatomy carrying the outline of a structure already destroyed. The protrusions identified as eyes were only nostrils. The real visual system had dissolved somewhere between the mesopelagic and the deck of the research vessel. Comb jellies and siphonophores suffer even more severely. Their bodies contain so little dense tissue that conventional net collection often tears them apart completely. A siphonophore extending through open water as a coordinated colonial structure becomes shredded gelatin and detached zooids tangled in mesh during retrieval. Delicate setanophores rupture under turbulence alone. For decades, scientific descriptions relied on fragments, isolated anatomical pieces, drifting tissue masses, or partially destroyed remains reconstructed after the fact. Entire behaviors remained invisible because the organisms themselves could not survive the act of observation. Phronima carried a similar confusion. Early specimens recovered from nets often appeared as damaged crustaceans trapped inside unidentified gelatinous debris. The transparent salt barrel surrounding the animal disintegrated under turbulence before retrieval reached the surface. Researchers saw the occupant without understanding the structure it had been living inside. The hollowing of the salp, the reshaping of the walls, the propulsion of the barrel through open water, all of it remained hidden until cameras observed the behavior directly in the mesopelagic itself. The transformation began slowly during the late 20th century as remotely operated vehicles descended into depths where fragile organisms could finally be watched alive. Equipped with cameras, manipulator arms, pressure-resistant housings, and controlled lighting systems, these vehicles altered deep-sea biology fundamentally. Observation no longer depended entirely on dragging bodies upward through kilometers of water. Animals could now remain inside the environment that shaped them, while science adapted to their conditions instead of forcing them into surface conditions they could not survive. During the 1980s, and increasingly through the 1990s and early 2000s, footage from research submersibles began revealing structures and behaviors that net collection had erased completely. In 2004, a remotely operated vehicle, operated by the Monterey Bay Aquarium Research Institute, documented the barreleye dome intact for the first time. Japetella's rapid transition from transparency to opaque red was filmed alive within the water column rather than inferred from damaged tissue. In 2020, the Ningaloo Canyons expedition captured the enormous spiral feeding posture of an Apolemia siphonophore extending through dark water in a form impossible to preserve physically. What emerged from these discoveries was not only new information, but an entirely different philosophy of study. Observe first, collect second. Sometimes do not collect at all. The implications remain difficult to overstate. Nearly everything confidently understood about transparent deep-sea animals has been learned through technologies only a few decades old. The century preceding them produced a scientific literature filled with partial truths assembled from bodies altered beyond recognition before reaching the surface. Entire anatomical structures vanished during ascent. Behaviors disappeared inside nets before anyone knew they existed. Even now, remotely operated vehicles visit only isolated regions within a mesopelagic realm, spanning roughly 1 billion cubic kilometers of ocean. Expeditions move through narrow corridors of darkness for limited periods of time, recording fragments from an ecosystem vastly larger than the observational tools passing briefly through it. Most of that volume has never been seen directly at all, and within it, transparent animals continue drifting through water that still resists being studied. For most of the 19th century, marine biologists believed they were studying a strange, transparent fish unlike anything else in the Atlantic Ocean. The animal drifted through open water as a thin, leaf-shaped body lined with oversized fangs, almost invisible except for the outline of its head and the narrow bands of muscle running through its sides. Researchers classified it as a separate species and gave it the name Leptocephalus brevirostris. It appeared so different from any known adult fish that no obvious connection seemed possible. Then, in the year 1896, the Italian zoologist Giovanni Grassi observed one of these transparent creatures changing form. The body shortened and thickened. Blood developed through tissues that had previously appeared almost empty. The head rounded. The fang-like teeth disappeared. What emerged was not a new species at all, but a juvenile eel. The drifting, transparent animal scientists had named separately for decades was only a temporary stage in the life of something already familiar. The leptocephalus is not a single species but a larval body form shared across more than 800 species of eel and eel-related fish, including moray eels, conger eels, and garden eels. The form itself emerged approximately 140 million years ago during the Cretaceous period and persists today across oceans worldwide, one of the most unusual developmental strategies in vertebrate life. Its anatomy appears almost unfinished. The body is laterally compressed into a transparent ribbon, thin enough that weak light passes through it almost entirely. Instead of dense muscle, the interior contains gelatinous material suspended beneath a narrow outer layer of segmented tissue. Red blood cells are absent during this stage of life, removing one of the strongest optical barriers inside vertebrate anatomy. The digestive tract remains little more than a simple transparent tube extending through the body. The teeth interrupt the illusion immediately. In the smallest larva, the fangs grow so large relative to the skull that the jaws cannot fully close around them. The mouth remains permanently open, lined with long, forward-pointing teeth extending outward into the water. They appear predatory despite the animal's fragility. Structures evolved for a life spent drifting through open ocean currents where prey encounters may be infrequent and brief. During metamorphosis, these teeth vanish completely. Adult eels develop an entirely different dentition suited for benthic life near reefs, coastlines, or seafloor burrows. The larval period varies enormously between species. Some leptocephali remain in open water for only a few months. Others drift for more than a year before transformation begins. European eel larvae travel farther still. Hatched in the Sargasso Sea, they drift thousands of kilometers across the Atlantic, carried largely by ocean currents alone, remaining transparent throughout a journey that can last as long as 3 years before reaching the coastlines of Europe. For much of the 20th century, no one understood what these larvae ate. Net-collected specimens consistently contained empty digestive tracts, and the transparency of the body complicated direct observation in the open ocean. The strongest current evidence suggests that leptocephali feed primarily on marine snow, microscopic organic particles descending slowly from upper waters. The diet leaves almost no identifiable remains inside the gut and produces no internal bioluminescent glow that would betray the transparent larva to predators nearby. As the journey nears its end, the transformation begins quietly. The long leaf-shaped body contracts inward, muscle thickens beneath the skin, blood fills the tissues. Pigment begins appearing gradually through the once transparent form. The fangs disappear. What remains is the glass eel, still translucent, but recognizably closer to the adult animal it will become. The change moves only in one direction, from invisibility toward visibility, from a body engineered for drifting through open water toward one built for life near the seafloor and coast. For the leptocephalus, transparency is temporary, a solution carried only through the earliest phase of life before another body replaces it entirely. Yet elsewhere in the mesopelagic, other animals remain transparent forever, preserving invisibility across entire lifespans, while the surrounding ocean grows darker and more optically dangerous with depth. At roughly 1,000 meters below the surface, the ocean changes its optical rules completely. The faint residual sunlight filtering downward through the twilight zone disappears at last, absorbed entirely by the water above. No ambient glow remains. No distant blue haze outlines drifting bodies from beneath. The long evolutionary pressure that shaped transparency through the upper mesopelagic begins weakening almost immediately because the silhouette problem itself no longer exists. Below this depth, light survives only where living organisms create it. Bioluminescence becomes the sole illumination of the environment, appearing as brief directed flashes generated intentionally by predators, prey, or mating animals moving through absolute darkness. Unlike sunlight, this light does not arrive diffusely from every direction. It appears in narrow beams, pulses, and controlled emissions, point source illumination produced by organisms carrying their own optical tools through the water column. Under these conditions, transparency changes from advantage to liability. When a bioluminescent searchlight strikes transparent tissue in complete darkness, the body no longer disappears into surrounding water. Instead, microscopic irregularities inside the tissue scatter the incoming light outward against a background of zero illumination. The animal glows where the beam touches it. A body that remained nearly invisible beneath weak ambient sunlight suddenly becomes outlined sharply by the very transparency that once concealed it. In response, many deep-sea animals abandoned transparency almost entirely as they moved deeper into the midnight zone. Another strategy emerged instead, one built not around transmitting light, but absorbing it completely. Ultra-black pigmentation evolved independently across several lineages of deep-sea predators and scavengers, including the Pacific black dragon and the fang-toothed fish. Their skin absorbs between 99.5 and 99.95% of incoming light, preventing meaningful reflection back into the surrounding water. A searchlight striking these animals returns almost nothing. No silhouette forms, no glare escapes. The body becomes a patch of missing information suspended within darkness. Between approximately 600 and 1,000 meters, however, the ocean occupies an unstable middle ground where both optical pressures remain active simultaneously. Weak ambient light still filters faintly downward from above, while bioluminescent searchlights sweep laterally through the water. This transition zone favors more flexible adaptations, and few animals embody that complexity more elegantly than Japetella. Transparent beneath ambient conditions, opaque red under directed blue illumination, the octopus survives precisely because it can respond to both forms of visibility at once. Other species divide the problem spatially across their bodies rather than switching states entirely. Ventral surfaces, facing downward toward deeper darkness, remain pale or partially transparent, reducing visibility from below against lingering background light. Dorsal surfaces facing upward develop darker pigmentation, capable of absorbing directed bioluminescent beams arriving from predators above. The animal carries two camouflage systems simultaneously, one aligned with the optical conditions beneath it and another with the conditions overhead. The red pigmented digestive systems discussed earlier remain useful throughout this depth range as well. Even where transparency fades, swallowed bioluminescent prey still threatens to illuminate the predator from inside. Dark stomach linings continue functioning as internal light traps, regardless of whether the outer body remains transparent, black, or somewhere between the two. What emerges across the mesopelagic and bathopelagic is not a single camouflage strategy, but a gradient tied directly to depth itself. Counter-illumination dominates where silhouettes remain dangerous. Dynamic transparency switching appears where both ambient light and searchlights overlap. Anti-reflective coatings reduce scatter within partially illuminated water. Ultra-black pigmentation takes over, where directed bioluminescence becomes the dominant visual threat. The ocean arranges these adaptations vertically through the water column with remarkable precision. Transparency, despite its extraordinary success, remains limited by the physics of light itself. It works best where weak illumination arrives broadly enough for bodies to vanish against it. Deeper down, darkness changes the equation, and other solutions emerge from the same evolutionary pressures. Yet even within those limits, the transparent zone extending from roughly 200 to 1,000 meters encompasses one of the largest inhabited spaces on Earth. An immense volume of water, filled with drifting bodies, evolved not merely to survive there, but to remain almost impossible to see. Far below the weather, below waves, below the sunlight that defines the familiar ocean, the mesopelagic zone spreads through the planet like a second atmosphere made entirely of water. It begins at roughly 200 meters depth and extends downward toward 1,000, occupying an estimated 1 billion cubic kilometers of space. By volume, it is the largest inhabited environment on Earth. Mountains, forests, deserts, grasslands, rivers, and coastlines together occupy only a fraction of the living space contained within this dim layer, suspended between the bright surface and the deeper midnight waters below. For most of human history, it appeared empty. The animals living there rarely broke the surface. Nets passing through the region returned damaged gelatinous fragments, transparent bodies dissolved into residue, or fish too delicate to survive a scent intact. Even now, the mesopelagic remains difficult to measure accurately because the animals inhabiting it are specifically adapted to avoid detection. Transparency, counter-illumination, distributed bioluminescence, rapid vertical movement, and fragile anatomy combine into an ecosystem that resists observation almost by design. Yet every modern estimate suggests extraordinary abundance. Conservative biomass calculations place the total mass of mesopelagic fish alone somewhere between 1 billion and 10 billion tons, potentially exceeding the combined biomass of all surface ocean fish known to commercial fisheries. The uncertainty itself reveals the limits of present knowledge. A factor of 10 separates the lowest and highest estimates, because traditional net surveys consistently fail to capture transparent species, fast-moving squid, delicate siphonophores, and gelatinous drifters that tear apart before reaching the deck. Sonar detects movement where nets retrieve almost nothing. The water column appears sparse to human eyes, while instrument readings suggest biological density on a planetary scale. Each evening, this hidden ecosystem rises. As darkness spreads across the surface ocean, immense numbers of lanternfish, glass squid, transparent crustaceans, salps, comb jellies, and siphonophores ascend hundreds of meters upward into shallower water rich with plankton and drifting prey. Before dawn, they descend again, returning to darker depths where visibility decreases and predators become less numerous. This migration repeats every day across every major ocean basin on Earth. Billions upon billions of animals moving vertically through the water column every 24 hours, a continuous planetary pulse occurring mostly beyond human sight. In terms of biomass and frequency, it is the largest animal migration known. The movement reshapes the chemistry of the planet itself. Animals feeding near the surface consume carbon fixed originally by phytoplankton through photosynthesis. When those animals descend, the carbon descends with them inside living tissue. Waste products sink farther. Dead bodies drift downward into deeper water and eventually toward the seafloor. Transparent mesopelagic organisms contribute heavily to this process because many gelatinous bodies sink rapidly once life ceases. Salps collapse into dense organic masses. Comb jellies and siphonophores descend as drifting carbon packages, moving nutrients away from the atmosphere for decades or centuries. Current estimates suggest the mesopelagic ecosystem may sequester between 1 billion and 5 billion tons of carbon each year. The uncertainty, again, remains enormous, because the animals performing the transport cannot yet be counted reliably across such a vast space. For a long time, this region was imagined as quiet, nearly empty water separating the productive surface from the abyss below. Acoustic recordings collected by remotely operated vehicles reveal something very different. Clicking, pulsing, feeding disturbances, muscular contractions, and signaling events fill the darkness continuously. Biological sound surrounds the instruments at nearly all depths. The twilight zone is not silent waiting space between ecosystems. It is an ecosystem itself, immense and crowded, and active beyond earlier assumptions. And despite containing one of the greatest concentrations of animal life on Earth, most of it remains effectively unexplored. Vast regions of the mesopelagic have never been visited directly even once. The migrations continue through water almost no human being has seen, inside an environment already beginning to change from warming oceans, industrial fishing pressure, and chemical shifts that began long before science understood what was living there. Across hundreds of millions of years, evolution repeatedly arrived at the same answer in the same place. Different animals, separated by immense spans of time and ancestry, entered the open ocean and slowly reshaped themselves toward invisibility. They did not share the same bodies, the same organs, or the same evolutionary history. Some were soft, gelatinous drifters, some were armored crustaceans, some were fish, squid, or colonial organisms composed of thousands of smaller living parts. Yet the environment surrounding them imposed the same demand with such consistency that unrelated lineages converged upon nearly identical optical outcomes. This process is known as convergent evolution. It occurs when unrelated organisms independently develop similar traits because they encounter similar environmental pressures. Wings emerged separately in birds, bats, and insects. Streamlined torpedo-shaped bodies evolved in fish, dolphins, and extinct ichthyosaurs because moving efficiently through fluid rewards the same geometry, regardless of ancestry. Camera-type eyes appeared independently in vertebrates and cephalopods, constructed through different developmental pathways, yet solving the same visual problem with striking similarity. In the open ocean, transparency became another example of this pattern. The pressure driving it was simple and relentless. In a water column without rocks, sediment, reefs, or vegetation, the body itself becomes the only visible object. Any surface reflecting or scattering light risks detection. Every lineage entering this environment encountered the same physical constraint. Every successful lineage responded by reducing its optical presence as much as biology allowed. The remarkable part is not merely that transparency evolved many times, it is that each group achieved it through entirely different biological machinery. Comb jellies, or sea ctenophores, evolved bodies composed almost entirely of water-rich mesoglea, reducing refractive boundaries until light passed through them with minimal disturbance. They belong to a separate animal phylum older than most complex marine predators alive today, with no close relationship to jellyfish despite superficial resemblance. True jellyfish and siphonophores belong instead to the cnidarians, an entirely different lineage that independently arrived at similar gelatinous transparency because the same environment rewarded the same optical disappearance. Salps followed another route entirely. Though visually closer to drifting jelly organisms, tunicates are more closely related to vertebrates than to either jellyfish or comb jellies. Their transparent barrel-shaped bodies emerged from separate anatomical foundations while converging upon the same solution. Cephalopods such as glass squid and Japetella retained complex nervous systems, active hunting behaviors, and muscular locomotion. Rather than becoming mostly water, they manipulated transparency through chromatophores. Pigment organs contracted so completely that the surrounding tissue became optically clear. Their invisibility depended not on simplicity, but on active biological control. Vertebrates reached transparency through still different mechanisms. The barreleye evolved a transparent cranial dome filled with fluid matched spectrally to seawater. Leptocephalus larvae reduced optical density by lacking hemoglobin-rich blood and replacing dense muscle with gelatinous filler. Transparent fish larvae, transparent skull structures, and transparent tissues appeared repeatedly despite the metabolic complexity of vertebrate bodies. Crustaceans, such as Phronima, developed transparent exoskeletons and anti-reflective coatings that suppressed light scatter from otherwise visible surfaces. Arthropod cuticle, normally rigid and opaque, was reshaped into something transmitting light instead of reflecting it. Different tissues, different developmental pathways, different chemistries, different ancestors, yet the same environment repeatedly narrowed possibility toward one dominant strategy. This reveals something important about the open ocean itself. Transparency there is not simply one camouflage option among many equally effective alternatives. In water without physical cover, where vision governs survival across enormous distances, transparency becomes the nearest thing evolution has to a universal answer. Every lineage entering the pelagic world encountered the same optical mathematics. Every successful lineage bent toward the same conclusion. And despite how often evolution discovered this answer, the animals shaped by it remained almost entirely unseen by the species attempting to study them. In cold Pacific water drifting between the surface and the deeper dark, a transparent jelly pulsed with brief green light long before anyone understood what the glow contained. Its body was delicate enough to vanish almost completely in its natural environment, a soft, gelatinous medusa, later identified as Achorea victoria. During the middle decades of the 20th century, the animal attracted the attention of the Japanese biologist Osamu Shimomura, who became interested not in its transparency, but in the chemistry responsible for its light. At the time, bioluminescence remained poorly understood at the molecular level. Researchers knew certain marine organisms could produce flashes of living light, yet the internal reactions driving the process were still being isolated species by species. Shimomura began collecting thousands of crystal jellyfish from Pacific coastal waters, carefully extracting luminous material from the thin ring structures surrounding the animal's bell. From these tissues, he isolated a protein later named aquarin, a compound that emitted blue light in the presence of calcium ions. But alongside aquarin, another protein appeared repeatedly during the purification process. This second molecule did not create light on its own. Instead, it absorbed the blue glow produced by aquarin and re-emitted it as green fluorescence. Shimomura named it green fluorescent protein, later shortened universally to GFP. Inside the jellyfish, the exact evolutionary purpose of the conversion remains uncertain. It may broaden the visibility of the flash, alter signaling properties, or shift wavelengths in a way more detectable to other organisms moving through the water column. The animal itself offered no explanation. It simply carried the protein within its transparent tissues as part of a biological system refined quietly in the open ocean. What mattered was what the protein allowed scientists to do. GFP possessed an extraordinary property. When exposed to blue or ultraviolet light, it glowed visibly green without requiring additional chemicals, substrates, or destructive preparation. More importantly, the gene encoding GFP could be inserted into the DNA of other organisms and attached directly to entirely unrelated proteins. Wherever those proteins moved, the fluorescence moved with them. For the first time, researchers could watch biological activity unfold inside living cells in real time without killing or dissecting the organism being studied. The effect on modern biology was immense. Proteins involved in cancer growth could be tracked through living tissue. Neurons firing inside active brains could be visualized through fluorescent signals spreading across networks of cells. Viral infections could be followed from the moment pathogens entered tissue to the stages of replication and spread. Developmental biologists could observe embryos forming organ systems cell by cell. Researchers studying neurodegenerative disorders could monitor how malformed proteins accumulated and propagated through nervous tissue over time. Gene therapy experiments could reveal precisely where inserted genes activated and where they failed. What had once been invisible inside living organisms became observable. Over the decades, GFP expanded into a massive family of related fluorescent proteins, engineered into different colors and sensitivities. Entire fields of molecular biology reorganized around imaging technologies made possible by the discovery. Hundreds of thousands of scientific studies eventually incorporated GFP-based methods in some form, turning a transparent marine animal into one of the foundational tools of modern biomedical research. In 2008, Osamu Shimomura, Martin Chalfie, and Roger Seen received the Nobel Prize in Chemistry for the discovery and development of green fluorescent protein. The award recognized not merely a laboratory achievement, but a transformation in humanity's ability to observe life itself. And the origin of that transformation remained suspended in the body of a drifting open ocean medusa, an animal so visually faint in its native habitat that most people passing through the Pacific above it would never know it existed at all. From within that nearly invisible body came one of the most important instruments science has ever possessed for illuminating the hidden machinery inside living things, a reminder that the deep ocean has often concealed discoveries long before humanity understood enough to recognize what it was looking at. The pressure arriving now from the surface ocean moves slowly enough that most people never see it happening. Temperature shifts by fractions of a degree. Chemistry changes molecule by molecule. Sediment drifts downward through water that once remained almost perfectly clear. Yet, for the transparent ecosystem suspended through the mesopelagic, these changes alter the physical conditions that shaped their bodies over evolutionary time. The twilight zone has never been isolated from the surface above it. Heat penetrates downward through ocean circulation, and over recent decades, measurable warming has expanded progressively into deeper water layers. For gelatinous animals whose tissues are composed almost entirely of water, temperature governs nearly every biological process. Metabolism, buoyancy regulation, reproductive timing, and migration behavior all respond closely to ambient conditions. A small thermal shift, experienced continuously across enormous regions, becomes biologically significant. SALPs already appear to be responding. In portions of the Southern Ocean and North Atlantic, bloom dynamics have begun changing in frequency and distribution. Regions once dominated seasonally by krill increasingly experience larger salp populations instead, altering the structure of food webs, extending upward toward fish, seabirds, and whales. Comb jellies have shown similar range adjustments, with populations gradually expanding toward colder polar waters as ocean temperatures rise. The transparent ecosystem is not disappearing uniformly. It is rearranging itself in motion. At the same time, seawater chemistry is changing. As atmospheric carbon dioxide dissolves into the ocean, it forms carbonic acid, lowering overall pH. The shift is subtle on human scales, yet profound for organisms dependent upon calcium carbonate structures. Among the most vulnerable are pteropods, tiny transparent sea snails drifting through upper mesopelagic waters. Their delicate shells are composed of aragonite, a crystalline form of calcium carbonate particularly susceptible to acidic dissolution. In sections of the Southern Ocean, living pteropods have already been observed carrying shells visibly eroded by surrounding seawater. The damage appears as pitted, thinning surfaces where structural material dissolves faster than the animal can replace it. These organisms occupy a critical position in open ocean food webs, consumed by fish, squid, whales, and numerous larger predators moving through the twilight zone. Their decline would not remain isolated to a single drifting species. It would propagate upward through the entire system surrounding them. Another pressure approaches not through chemistry, but through industry. Across the Central Pacific, the Clarion-Clipperton Zone has become a major target for proposed deep-sea mining operations, seeking polymetallic nodules rich in nickel, cobalt, copper, and manganese. The region overlaps directly with mesopelagic habitats populated by transparent organisms adapted specifically to exceptionally clear water conditions. Mining activities do not disturb only the seafloor itself. Sediment plumes generated by collection systems can rise upward into the water column, introducing suspended particulate matter into regions where optical clarity governs survival. For transparent animals, suspended particles alter the environment physically as well as visually. Counter-illumination systems rely upon precise background matching. Photophore signaling depends on predictable light transmission through water. Refractive index camouflage functions best when surrounding water remains optically stable. A water column filled with artificial particulate haze changes the conditions these organisms evolved within over millions of years. During a 2023 environmental survey within the Clarion-Clipperton region, researchers documented a previously unseen squid behavior. The animal buried itself partially within soft seafloor sediment, exposing only its tentacles while mimicking surrounding organisms. Whether the behavior functioned for hunting, camouflage, or both remained uncertain. The species itself was unidentified. The observation occurred not during a dedicated biological expedition, but during industrial impact assessment work preceding potential extraction activity. That detail carries its own quiet weight. For most of human history, transparent mesopelagic animals remained effectively invisible to science. Their populations were never comprehensively measured before environmental pressures began altering the ecosystems surrounding them. Detecting decline requires accurate baselines, and accurate baselines for invisible organisms have existed only briefly. Loss becomes difficult to quantify when the living system was never fully counted in the first place. The ocean does not preserve unknown things simply because they remain unseen. In many cases, invisibility delays recognition until the changes are already underway. Every transparent animal drifting through the twilight zone carries, written into its body, a record of the eyes that evolved beside it. The glass squid erases its outline because predators below search the dim surface glow for silhouettes. Japetella changes from transparent to red because hunting fish sweep the water with blue bioluminescent light. Phronima dampens reflections from its exoskeleton because scattered light has become a detectable signal in the darkness around it. Each solution exists because another animal learned how to see. None of these camouflage systems developed in isolation. They emerged through interaction, through millions of years of visual negotiation between hunter and hunted. Transparency is not simply a characteristic, like size or color. It is a relationship between one organism and the sensory system attempting to locate it. A glass squid drifting through open water may vanish almost completely against the surrounding sea when viewed by a hatchetfish below. The same animal becomes immediately obvious under the concentrated illumination of an ROV camera descending from the surface world. Under polarized light, different structures appear again, revealing boundaries, invisible under ordinary observation. The squid itself has not changed. Only the observer has changed. This remains true across the entire mesopelagic ecosystem. The barreleye's dome functions specifically within the spectral conditions of faint downwelling sunlight. Counter-illumination works only because the photophores beneath an animal reproduce the exact intensity and wavelength of the light above it. Ultra-black fish absorb nearly every photon because bioluminescent searchlights transformed darkness into a directional hunting environment. Every adaptation described throughout this ocean exists relative to a particular form of perception. Visibility in the deep sea is never absolute. It exists between observer and observed, between biological signal and biological interpretation. The same organism may be simultaneously present and absent, depending upon the sensory architecture examining it. To one predator, it dissolves completely into background light. To another, using a different wavelength or viewing angle, it becomes detectable immediately. This is not metaphor, it is ecology expressed through optics. For most of human history, the mesopelagic zone appeared sparse because the tools used to study it were designed for surface conditions. Nets destroyed gelatinous bodies before they reached daylight. Sonar missed transparent tissue. Cameras illuminated animals with wavelengths that altered natural behavior, or overwhelmed camouflage systems evolved for darkness. The zone itself remained unchanged while the instruments describing it remained incomplete. Only after submersibles and remotely operated vehicles began entering the environment directly did the apparent emptiness dissolve. Suddenly, the water column revealed layered migrations, transparent predators, drifting colonies, light signals, feeding structures, and complex behaviors unfolding continuously through depths once assumed nearly barren. The ocean had not transformed into a richer place. Human observation had finally adjusted itself to the conditions already there. The implication extends beyond the animals themselves. 1 billion cubic kilometers of inhabited ocean remained functionally hidden because perception depended upon tools mismatched to the environment being studied. The absence existed partly inside the instrument. What appeared empty reflected the limits of the observer as much as the reality being observed. Transparent animals expose this limitation with unusual clarity because their survival depends upon manipulating perception directly. They endure by exploiting assumptions built into visual systems. They disappear not because they cease existing, but because another organism lacks the capacity to distinguish body from water, signal from background, structure from surrounding space. In the twilight zone, invisibility is never complete. It is conditional, temporary, and observer-dependent. Every animal there lives inside a shifting field of detection where appearance changes with wavelength, depth, angle, movement, and biology. The mesopelagic ocean itself may represent the same principle at planetary scale. For centuries it seemed distant, empty, and biologically thin because humanity possessed no reliable way to perceive it properly. Once the correct instruments arrived, an ecosystem emerged from the darkness that had been present the entire time, moving silently through water beneath the limits of ordinary sight. What appears absent sometimes remains only unrecognized by the way we are looking. A siphonophore drifting through the mesopelagic ocean does not behave like any familiar animal because, in a strict biological sense, it is not built like one. Every section of its body is alive. Every section is genetically identical. Every section originated from a single fertilized egg, dividing and specializing into different forms until the colony became a chain of dependent organisms connected into one continuous structure. Some zooids pulse rhythmically to generate movement through the water. Others trail long curtains of tentacles armed with nematocysts. Others digest captured prey. Others reproduce. Each zooid performs only one task, permanently specialized for a single role. None can survive independently for long. Removed from the colony, they cease functioning as viable organisms. Connected together, they become something larger that behaves with extraordinary coordination despite possessing no central brain and no singular controlling body. The siphonophore, therefore, occupies an unstable place in biology itself. A jellyfish can be identified as an individual animal. A fish can be identified as an individual animal. A siphonophore resists the category entirely because it exists simultaneously as many organisms and as one integrated entity. The colony swims, feeds, grows, repairs itself, and reproduces as a single system while remaining physically composed of thousands of living units, each technically alive on its own terms. This becomes more difficult to define as the colony grows larger. The Apolemia siphonophore, filmed during the Ningaloo Canyons expedition in 2020, measured an estimated 119 meters if fully extended. Suspended through open water, nearly transparent against the surrounding sea. It maintained coordinated feeding structures across a distance longer than a football field. Electrical signals traveled through the colony. Tentacles responded to contact. Swimming zooids adjusted movement. Digestive zooids processed prey. Reproductive zooids continued their own functions simultaneously. No single part understood the whole. No zooid perceived the full geometry of the feeding spiral drifting through darkness. No zooid decided where the colony would move. No zooid experienced the capture of prey as a unified event. Yet the colony captured prey. The colony adjusted its structure. The colony survived. For millions of years, siphonophores have persisted in this form, drifting through open water as distributed organisms whose intelligence exists not in a brain, but in the interaction between specialized living components. Their coordination emerges from arrangement itself, from chemical gradients, electrical signaling, and inherited developmental rules operating continuously across bodies connected into a larger biological architecture. Death, when applied to such a system, becomes difficult to locate precisely. If a shark loses half its body, the shark dies. A siphonophore may lose enormous sections of its colony and continue functioning. Zooids are damaged, replaced, regenerated, and reorganized over time. Portions detach. New sections develop. The colony changes shape continuously throughout its existence, while remaining recognizably the same entity. At what moment, then, does the colony stop being itself? Biology contains definitions for individuality, but siphonophores strain against all of them. They are genetically unified, yet physically divided. They behave collectively, yet possess no central awareness. They are composed of organisms that cannot survive alone, yet are themselves alive. The deeper implication emerges slowly from the water around them. Human categories for life were constructed around visible animals living in familiar environments, creatures with singular bodies, singular nervous systems, singular deaths. Mammals, birds, reptiles, fish, organisms shaped in ways surface observation could easily separate and define. Transparent deep ocean animals repeatedly resist those boundaries. Comb jellies resemble jellyfish despite evolving separately. Transparent larvae appear unrelated to the adults they become. Colonial siphonophores exist somewhere between organism and ecosystem. The farther observation moves into the open water column, the less stable ordinary definitions appear. The problem is not with the animals themselves. The siphonophore is not confused about what it is. The confusion belongs to the observer, attempting to force a surface world vocabulary onto forms of life that evolved in an environment where visibility itself became uncertain. Long before vertebrates entered the ocean, before fish developed jaws, before trilobites crossed the seafloor beneath the first complex ecosystems, soft, transparent bodies were already drifting through ancient water. The oldest oceans on Earth were inhabited by organisms that sensed their surroundings without truly seeing them, responding instead to chemistry, vibration, pressure, and movement carried through seawater in darkness older than vision itself. The first recognizable eyes appear in the fossil record approximately 540 million years ago during the Cambrian explosion, when animal life diversified with extraordinary speed into increasingly complex forms. Light-sensitive patches became directional organs. Directional organs became primitive, image-forming eyes. Once organisms could detect shape and movement with precision, the ocean changed permanently. Predators became hunters in a new sense. Prey became visible objects against their surroundings. Evolution accelerated into an arms race. driven partly by perception. Yet, many transparent body plans already existed before this transformation began. Comb jellies have occupied the oceans for more than 500 million years. Early siphonophore ancestors emerged hundreds of millions of years ago as colonial drifting organisms suspended through open water. Gelatinous tunicates and primitive soft-bodied planktonic forms were already exploiting the optical properties of water-rich tissue long before complex visual systems spread across marine ecosystems. This alters the meaning of transparency itself. In the modern Mesopelagic Ocean, transparency appears closely tied to camouflage against visually hunting predators. Glass squid dissolve against faint background light. Transparent larvae drift unnoticed through open water. Counter-illumination erases silhouettes produced by bodies crossing dim sunlight from above. Every transparent organism now exists inside a world shaped by eyes. But transparency did not originate inside that world. The earliest selective pressures favoring gelatinous, low-density, water-rich bodies were likely unrelated to vision entirely. Before image-forming eyes became widespread, predators located prey through chemical gradients, direct contact, fluid disturbances, and pressure changes traveling through seawater. A body composed largely of water may have produced weaker turbulence. It may have blended chemically with the surrounding environment more effectively than dense muscular tissue. Soft transparency may initially have been advantageous simply because it reduced detectable difference between organism and ocean. Then vision emerged. Once eyes spread through marine ecosystems during the Cambrian period, existing transparent bodies acquired a second advantage their ancestors had never evolved specifically to possess. A form already suited to drifting through water became difficult to see within it. Gelatinous tissue, originally shaped by ancient non-visual pressures, entered an ocean increasingly organized around detection by sight. Transparency was not invented in response to eyes. It was inherited into a world that had learned how to look. From that point onward, evolution refined the strategy continuously. Refractive indices shifted closer to seawater. Pigments disappeared. Chromatophores contracted permanently. Blood chemistry changed. Optical scattering diminished across tissues and organs. Transparent animals became more specialized with every geological age, shaped simultaneously by ancient physical constraints and increasingly sophisticated visual predators. The deep ocean still preserves traces of that earlier planetary condition. Far below the surface, pressure dominates structure. Sunlight disappears. Chemical gradients regain importance. Bioluminescence replaces daylight with isolated bursts of controlled illumination. Sound, vibration, and dissolved molecules travel enormous distances through darkness. In some respects, the Mesopelagic and Bathypelagic zones resemble fragments of a more ancient Earth, environments where information moves through water in ways that do not rely entirely on ordinary sight. The transparent animals living there are not primitive survivors from a forgotten world. Their biology is extraordinarily refined, calibrated through hundreds of millions of years of evolutionary adjustment. Yet the foundation beneath that sophistication remains genuinely ancient. A comb jelly drifting through darkness carries within its body a lineage older than most complex marine predators. A glass squid hovering beneath the twilight zone represents a modern expression of optical strategies rooted in bodies that existed before vertebrates, before forests, before terrestrial ecosystems, before eyes themselves had fully transformed the sea. The ocean learned to see after transparency already existed. At midday, standing above the open ocean, the visible world appears remarkably simple. Sunlight scatters across moving water. Reflections fracture and reform with the wind. Beneath the surface, there is usually nothing to anchor the eye. No reef structure, no coastline, no indication that an enormous inhabited space begins immediately below the thin reflective boundary separating atmosphere from sea. The illusion of emptiness exists because almost everything living there has evolved specifically to preserve it. Below the surface extends approximately 1 billion cubic kilometers of inhabited water, a volume larger than any ecosystem humans experience directly. Through this space move lanternfish, transparent crustaceans, comb jellies, salps, siphonophores, glass squid, larval eels, drifting colonies, and soft, gelatinous predators that collectively may represent one of the largest concentrations of animal biomass on Earth. Every night, billions ascend toward the surface to feed. Every dawn, billions descend again into dimmer water below. Carbon moves with them, nutrients move with them, energy moves with them. Atmospheric chemistry is influenced partly by migrations occurring silently through darkness. Most human beings will never enter. Yet from above, the ocean appears nearly empty. The mesopelagic zone is not hidden merely because it exists at depth. Mountains on land remain visible from great distance, despite scale and remoteness. The twilight zone remains obscure because the organisms inhabiting it evolved under relentless pressure to reduce detectable information about themselves. Light passing downward through their tissue is transmitted rather than reflected. Bioluminescent signatures are canceled by counter-illumination. Shadows are erased. Internal organs are concealed behind dark pigmentation. Refractive boundaries are minimized until body and seawater become optically similar. The result is an ecosystem built from reduced signal. A glass squid, suspended 700 meters beneath the surface, is not attempting to disappear from scientific observation. It evolved transparency because predators beneath it learned to search for silhouettes against faint ambient light. A siphonophore drifting through open water does not conceal itself intentionally from human cameras. Its gelatinous structure simply produces very little optical contrast against the surrounding sea. The same biological solutions that defeat hatchetfish, dragonfish, and lanternfish happen also to defeat surface-based observation systems designed by another species entering the environment much later. This distinction matters. The word hidden suggests secrecy, intention, or deliberate concealment. Transparent deep ocean animals possess no concept of secrecy. They do not know they are difficult to observe. They simply exist in forms shaped by evolutionary pressures unrelated to humanity. Their invisibility is a consequence of physics and biology interacting across immense periods of time. The ocean itself is similar. It is not withholding information. Vast regions of it simply contain organisms that return almost no useful signal to instruments built originally for the bright, particulate-rich conditions of surface water. Nets destroy fragile transparency before specimens reach daylight. Sonar passes through gelatinous tissue weakly. Cameras overwhelm natural optical environments with artificial illumination. For centuries, scientific absence emerged, partly from methodological mismatch between observer and habitat. When remotely operated vehicles finally entered the twilight zone directly, the apparent emptiness dissolved into movement and structure. Colonies longer than whales drifted through darkness. Transparent octopuses altered their bodies in response to blue light. Fish, with fluid-filled skull domes, rotated tubular eyes toward prey, silhouettes invisible to human vision. Entire migrations unfolded daily beneath shipping lanes and weather systems unnoticed from above. The ocean did not suddenly become inhabited. Humanity simply acquired instruments capable of perceiving a fraction of what had always been there. The mesopelagic zone therefore represents a particular kind of unknown space, not empty but undetected, not distant but optically inaccessible. Presence exists there continuously, without announcing itself. Life moves through the water without leaving strong visual evidence that movement occurred. From the surface, the open ocean appears blue, reflective, and almost featureless. Beneath that appearance drifts one of the largest living systems on Earth, composed substantially of animals whose greatest evolutionary achievement has been learning how not to be seen. 700 meters beneath the surface, Japetella drifts through water so dark that the remaining sunlight resembles memory more than illumination. Its body hangs almost motionless in the mesopelagic current, soft tissue transmitting the dim blue glow filtering down from far above. A hatchetfish scanning upward for silhouettes detects nothing. The outline that should interrupt the water is absent. The octopus remains present physically, metabolically active, fully alive, yet optically unresolved against the surrounding sea. Then another signal enters the water. Somewhere nearby, a lanternfish sweeps a concentrated beam of bioluminescent blue light through the darkness, searching for transparent tissue that might scatter its illumination back toward the predator's eyes. Japetella detects the wavelength almost immediately. Chromatophores expand across its body in less than a second. Transparency disappears. The body darkens into deep red, absorbing the incoming blue light before reflection can occur. The searchlight passes over the water column. The octopus remains undetected again. Moments later, the light is gone. Chromatophores contract. Transparency returns. Throughout the entire sequence, the animal never ceased existing. It did not vanish physically. Water continued moving around its body. Nerves continued transmitting signals. Muscles adjusted posture. Sensory systems monitored the surrounding environment continuously. Yet for most nearby organisms, Japetella occupied the ocean as a kind of controlled absence, visible only during brief intervals when conditions aligned in exactly the wrong way. Its existence depends upon remaining outside the perceptual world of the animals around it, not hidden behind rocks or buried beneath sediment, not concealed through distance, present directly within shared open water while transmitting so little usable optical information that other organisms fail to register it as a body at all. The distinction matters because transparency in the mesopelagic ocean is not symbolic. It is literal physics acting through living tissue. Light passes through Japetella rather than scattering strongly from its surface. The animal reduces its own detectable boundary with the surrounding environment until predator visual systems interpret the space it occupies as empty water. And still, the animal lives there. It hunts. It reproduces. It navigates pressure gradients and chemical signals across a water column, spanning enormous distances in every direction. It participates fully in an ecosystem that often cannot perceive it clearly while it moves through the same shared environment as predators, prey, drifting colonies, and migrating fish. There is something difficult to stabilize intellectually in that condition. Surface animals, including humans, evolved in environments where existence and visibility remain closely linked. Bodies occupy space visibly. Movement generates obvious disturbance. Presence announces itself continuously through reflected light, sound, texture and shadow. Even camouflage on land usually depends upon resemblance to something already visible: bark, sand, leaf litter, stone. Japetella approaches a different condition entirely. Its body does not imitate the surrounding environment. Its body approximates the optical properties of absence itself. The ocean flows around and through its transparent tissues with minimal interruption to the surrounding light field. Nearby predators do not ignore it consciously. They frequently fail to detect it at all. Biology explains how this works. Refractive indices, chromatophores, spectral sensitivity, counter-illumination, scattering reduction, these mechanisms can be measured precisely. The deeper implication remains more difficult to contain within technical language. An organism can exist fully inside an environment while remaining largely excluded from the perceptual reality of the other organisms sharing that environment. Presence and detection are not identical states. The mesopelagic ocean demonstrates this continuously through living bodies drifting beneath the surface world. What experience means for Japetella itself cannot be answered. Consciousness in another organism remains inaccessible, even in familiar animals. In a transparent octopus suspended through darkness, the question becomes stranger still. Its sensory world, whatever form it takes, unfolds inside an ecosystem where survival depends upon reducing evidence of existence to the lowest possible level. And somewhere beneath the Pacific tonight, while currents move silently through cold open water, Japetella continues doing exactly that. Transparent, active, alive beyond visibility. While far above the surface, another species sits in darkness, trying for a moment to imagine it. Long before remotely operated vehicles drifted through the mesopelagic, with stabilized cameras and precision lighting, the deep ocean revealed itself only in fragments: torn tissue in nets, gelatinous residue collapsing in collection jars, structures destroyed by pressure change before they reached the deck. For most of scientific history, transparent animals entered human knowledge already damaged, already altered by the act of finding them. And yet the discoveries still came. Not in straight lines, not through perfect planning, more often through patience, coincidence, and the willingness to keep looking at places that initially appeared empty. In the 1960s, the biochemist Osamu Shimomura collected thousands of crystal jellyfish from Pacific waters because he wanted to understand how bioluminescence worked inside their transparent tissue. The goal was specific and technical: isolate the molecules responsible for the glow, study the chemistry. Inside the jellyfish, he identified aquarin, the compound producing blue light. But alongside it was another protein, green fluorescent protein, absorbing one wavelength and re-emitting another. The second protein was not the target of the research. It emerged from the work almost incidentally, a discovery nested inside another discovery. Decades later, that transparent medusa would become part of modern medicine, genetics, cancer research, and cell biology, all because someone had become curious about why a nearly invisible animal glowed in the dark. Far below the Pacific surface, another misunderstanding persisted for more than a century, because the evidence reaching scientists was incomplete from the moment it arrived. Barreleye fish collected in nets appeared to possess exposed tubular eyes and an oddly featureless forehead. The transparent dome enclosing the eyes collapsed during ascent, leaving only damaged remains behind. Then, in 2004, researchers at the Monterey Bay Aquarium Research Institute sent an ROV through mesopelagic waters and filmed the fish alive. The dome was there the entire time, fluid-filled, clear, enclosing the rotating eyes like a transparent shield. The correction of a century-old error arrived not through a new theory, but through finally seeing the animal before the ocean and pressure change destroyed the structure being studied. The same pattern continued elsewhere in the dark. In 2020, the Ningaloo Canyons expedition surveyed biodiversity off Western Australia. Cameras moved slowly through open water recording, drifting life forms, suspended particles, darkness between brief encounters. Then the footage revealed a giant Apolemia siphonophore coiled into a feeding spiral, stretching across a distance longer than a football field when fully extended. The largest living organism ever recorded had not been deliberately searched for. It appeared because cameras happened to be present while the colony occupied that exact configuration in open water. 3 years later, another survey descended into the Clarion-Clipperton zone to assess possible mining impacts on the seafloor. The purpose was industrial environmental evaluation. During the observations, cameras recorded an unidentified squid burying itself in sediment with only its tentacles exposed, mimicking surrounding organisms in a behavior never previously documented. The species itself remained unnamed. Again and again, the deep ocean has revealed itself this way, not through command but through encounter. The transparent animals were never absent during the centuries when science failed to describe them correctly. Phronima was already moving through the water inside its hollow barrel. Glass squid were already hovering motionless beneath fading sunlight. Comb jellies were already drifting through cold currents on bodies refined across 500 million years. The siphonophores were already extending invisible feeding structures through the mesopelagic dark. Human understanding arrived afterward, slowly, carrying instruments that first shattered, collapsed, or overlooked the very things they were built to study. Only later did the tools become delicate enough to enter the ocean without forcing it immediately to disappear. Even now, after decades of deep-sea exploration, the actual territory directly observed by human instruments remains astonishingly small. The remotely operated vehicles drifting through the twilight zone, the research submersibles lowering cameras into cold water, the fragile specimens collected intact enough to analyze, all of them together illuminate only scattered points inside a habitat, so vast it resists comprehension. The mesopelagic zone stretches across the planet beneath nearly every ocean basin, occupying roughly 1 billion cubic kilometers of water between the sunlit surface and the true abyss below. Within that volume exists one of the largest concentrations of animal life on Earth. Yet, the scientific measurements describing that life still carry uncertainties so large they would be unacceptable almost anywhere else in biology. Current estimates for twilight zone fish biomass range from 1 billion tons to 10 billion tons. The upper estimate is 10 times larger than the lower one. 1,000% uncertainty attached not to a minor population but to one of the largest ecosystems on the planet. The reason is not carelessness, it is visibility. Traditional net sampling, the method used for most of the history of oceanography, performs badly in the mesopelagic. Fast-moving species detect approaching nets through pressure waves and escape. Gelatinous bodies rupture during capture. Transparent organisms pass through mesh openings or collapse into residue, too damaged to identify properly. The animals least likely to survive collection are often the same animals appearing in the greatest numbers on sonar. So science learned to listen instead. Across the oceans, acoustic instruments map the deep scattering layer as a moving band of density rising each night toward the surface and sinking again at dawn. Sonar confirms enormous quantities of life suspended in the water column. It measures echoes returning through darkness from bodies no human eye has directly seen. But sonar is imprecise in a different way. It reveals presence without identity. A dense acoustic layer may contain lanternfish, glass squid, salps, siphonophores, comb jellies, shrimp, transparent larvae, or species that exist nowhere in the scientific record beyond an unexplained signal in the water column. Sonar cannot separate one drifting organism from another when all are compressed into a shifting acoustic mass. It confirms abundance while withholding detail. Direct visual observation remains limited to a small number of repeatedly studied locations. Monterey Canyon, off California, waters surrounding Hawaii, sections of the Azores, isolated transects in the Southern Ocean, the Arabian Sea, and several deep basin sites reachable by institutions wealthy enough to operate advanced submersible systems. These regions appear frequently in scientific literature, not because they represent the ocean broadly, but because they are among the few places where sustained observation has been technologically possible. Outside those narrow corridors lies most of the mesopelagic world. Water columns, thousands of meters deep, extending across entire ocean basins, have never been continuously filmed. No instrument has lingered there long enough to document the full rhythm of life moving through them. Vast migrating populations pass nightly between darkness and surface waters, beyond the range of direct human observation, carrying carbon, reproducing, hunting, glowing, disappearing again before dawn. Within that immensity, there are almost certainly transparent animals never formally described, not hidden rarities occupying isolated caves or hydrothermal vents, but ordinary inhabitants of open water existing in populations large enough to shape ecosystems while remaining scientifically unnamed. Creatures that no intact specimen has reached the surface to represent, bodies seen briefly on damaged footage without taxonomic certainty, organisms registered acoustically but never visually, forms passing through darkness outside the narrow cones of artificial light produced by descending cameras. The deep ocean contains entire categories of absence created not by emptiness, but by limitation in the instruments attempting to observe it. And so the strange situation remains. Somewhere tonight, through billions of cubic kilometers of cold water, transparent animals are drifting, feeding, reproducing, and vanishing against the darkness around them, not metaphorically unseen, literally beyond the reach of every camera, net, and submersible ever built, existing in numbers large enough to move planetary systems while remaining almost entirely outside human sight. Far below the moving weather of the surface, beneath storms, shipping lanes, coastlines, and human borders, the deep ocean continues in conditions older than animal life itself. The darkness was there before eyes evolved to perceive it. The pressure existed before anything living possessed tissue strong enough to endure it. Cold water circulated through the planet's basins for billions of years before the first transparent body drifted through it. The ocean did not change itself to receive life. Life entered an environment already ancient. Salt chemistry, density gradients, dissolved minerals, slow thermohaline circulation, moving between polar seas and abyssal plains. All of it predates the creatures now suspended inside it by spans of time so large that human history disappears completely within them. Even the continents above have rearranged themselves repeatedly while the deep basins remained dark and under pressure below. Then, eventually, animals arrived. The transparent forms moving through the mesopelagic today are not recent experiments. Comb jellies have existed in some recognizable form for more than 500 million years. Their drifting bodies, composed almost entirely of water, already occupied the oceans before vertebrates emerged onto land, before forests spread across continents, before flowering plants altered the chemistry of the atmosphere, before insects filled the air. Siphonophores, too, belong to lineages reaching back through immense geological spans, colonial bodies refining their distributed existence across eras during which entire groups of surface animals appeared and vanished. Long before mammals evolved, long before primates, these transparent organisms were already solving the same environmental problem they solve now: how to remain undetected in open water where there is nowhere solid to hide. The ocean surrounding them changed continually through those ages. Temperatures shifted, oxygen levels rose and fell, extinction events erased dominant species and reorganized ecosystems, continents separated, ice advanced and retreated across the poles, new predators evolved eyes sensitive enough to detect silhouettes against dim light from above, bioluminescent searchlights emerged in response, counter-illumination followed. Transparent tissues became darker when illuminated directly. The details changed constantly, but the central condition remained remarkably stable. Open water offered no shelter, and so the same solution persisted across hundreds of millions of years. Transmit light rather than reflect it. Reduce the body's optical signature until the surrounding ocean passes through tissue almost uninterrupted. Become difficult to register in an environment where detection means death. These animals have been refining that strategy across spans of evolutionary time so vast that the existence of human beings occupies only the final fraction of the story. Modern humans emerged roughly 300,000 years ago. Industrial civilization is only centuries old. Cameras capable of descending intact into mesopelagic darkness have existed for mere decades. Yet now, for the first time in the history of either species involved, one animal is observing the other alive. A transparent comb jelly drifting through darkness beneath a remotely operated vehicle represents an encounter between evolutionary histories separated by almost unimaginable scales of time. One lineage refined invisibility over 500 million years. The other discovered electricity, optics, pressure-resistant housings, and digital imaging quickly enough to lower artificial eyes into the deep before fully understanding the ecosystem waiting there. The camera itself is young enough that many people alive today are older than the technology enabling these observations. The animal being filmed is older than most species that have ever existed on Earth. And still it drifts there almost unchanged in purpose, moving through dark water on a body plan refined across geological eras, while somewhere above, surface creatures suspend lights and lenses into the depths trying, briefly, to look at something that evolved for almost the entire history of complex life, specifically not to be seen. Beyond that point, the language of measurement begins to thin. The depths remain what they always were: cold, dark, pressurized, and largely indifferent to whether anything above the surface understands them at all. 700 meters below the surface, the ocean settles into conditions almost no human body will ever experience directly. The temperature holds near 4 degrees Celsius. Pressure compresses every exposed surface with a force roughly 70 times greater than the atmosphere above the waves. Sunlight no longer arrives intact. What little remains has already been filtered through hundreds of meters of water, leaving behind darkness interrupted only by the brief signals animals create for themselves. In this water, something transparent is moving now, not in theory, not as reconstruction or metaphor. A real body is drifting through the mesopelagic at this moment, transmitting light through tissue engineered by evolution to leave as little optical trace as possible. Photons pass into one side of the animal and emerge from the other almost unchanged. There is scattering, but very little, reflection, but rarely enough to matter. To the eyes around it, the body approaches absence closely enough that detection systems refined across millions of years often fail to register it at all. A glass squid may hover almost motionless between layers of dim blue light and deeper darkness, adjusting tiny movements through gelatinous fins while the water carries it slowly through open space. Nearby, a Japetella drifts transparent until the spectral signature of blue bioluminescence triggers pigment cells beneath its skin. Somewhere else, Phronima moves inside the hollow body of a salt barrel, suspended in currents older than recorded history. None of them know they have become objects of study. The names attached to them exist only on the surface, carried in human language, stored in databases and scientific papers, and whispered documentaries played in dark rooms far above the habitat itself. Japetella diaphana, Cranchiidae, Phronima sedentaria. These sounds mean nothing in the water where the animals live. The animals navigate through gradients of pressure, chemistry, motion, and light without any awareness that another species has begun lowering cameras toward them. They detect predators not through abstract understanding, but through wavelength, vibration, shadow, chemical trace. A lanternfish searchlight sweeps the water column. Chromatophores respond. A feeding tentacle brushes transparent tissue. Escape muscles contract instantly. Life proceeds through reactions refined over spans of time longer than human civilization can easily comprehend. And yet the most unusual thing about these animals may not be their transparency itself, but the duration of it. Bodies like these, or close ancestral versions of them, have existed in Earth's oceans for hundreds of millions of years, long before mammals appeared, long before forests covered continents, long before there were nervous systems capable of reflecting on invisibility as an idea. The ocean already contained organisms moving silently through open water on bodies designed to avoid detection. There was no audience for them then, no observer standing outside the system considering what it meant for a living thing to disappear optically into its environment. The transparent animals simply existed because the ocean selected for bodies that returned less information to predators around them. Even now, the animals themselves remain untouched by the concepts imposed upon them. They are not lonely in the darkness. Loneliness requires forms of self-awareness and social cognition that many of these organisms almost certainly do not possess. They are not mysterious to themselves. They are not hidden intentionally. They are simply alive in conditions where transmitting light more efficiently than reflecting it became one of evolution's most successful strategies. The deep ocean continues holding them exactly as it held their ancestors. Cold water moving slowly beneath the surface world, pressure compressing gelatinous tissue into functional forms. Darkness interrupted by flashes of bioluminescence, carrying signals between animals that rarely see one another clearly. Invisible bodies feeding, reproducing, drifting, dying, and returning their material back into the surrounding sea. The ocean has contained this hidden ecology longer than complex life has existed on land. It still does. And somewhere beneath the surface tonight, beyond the reach of sunlight and far beyond ordinary sight, transparent animals continue moving through the dark exactly as they always have. At the beginning of this journey, the expectation was simple: deep water, strange bodies, monsters waiting in the dark beneath the surface world. And the ocean did offer creatures that, at first glance, seemed to justify the word. Phronima moving through open water inside the hollowed body of another animal, carrying the transparent barrel like a drifting chamber through the mesopelagic dark. The siphonophore, extending itself across distances longer than most whales, a colonial organism composed of thousands of smaller living parts functioning together without any central awareness. The glass squid hovering almost invisibly in cold water, its transparent body interrupted only by a dark red digestive organ suspended at the center like a floating wound. Seen briefly under artificial light, these animals appear alien because the environment shaping them is so distant from ordinary human experience. Pressure 70 or 80 times greater than the atmosphere above. Water cold enough to slow nearly every metabolic process. Darkness interrupted only by the light living things create themselves. But the longer the ocean is observed, the less useful the word monster becomes. Monster suggests something opposed to us, threatening us, built in contrast to human life. These animals are not organized around humanity in any way. They do not know we exist. Their adaptations were not shaped against us. They are predators and prey only within the narrow ecology surrounding them, hunting and avoiding one another through wavelengths, silhouettes, chemical traces, and movement in water. No human body could survive without machinery. What they are, more accurately, is present. Completely present. Present in an ocean crossed constantly by ships and aircraft, measured by satellites, mapped by trade routes, depended upon for climate regulation, food systems, oxygen production, and weather patterns, while the immense living world beneath the surface remained largely unseen for almost the entirety of human history. The animals are still there now, somewhere below the reach of sunlight, perhaps 800 meters down. A glass squid is hovering almost motionless between layers of water, maintaining neutral buoyancy in darkness, while faint bioluminescent flashes drift around it. Japetella moves through the twilight zone, transmitting light through transparent tissue until the blue wavelength of a hunting searchlight forces its body temporarily into red opacity. Phronima continues, propelling its barrel through open water in the Atlantic, compound eyes scanning the dark for another gelatinous body large enough to reshape into shelter. None of this pauses when human attention turns elsewhere. The transparent world continues independently, uninterrupted by whether cameras descend into it or remain at the surface. The migrations still rise each night toward shallower water. Siphonophores still extend invisible feeding structures through passing currents. Comb jellies still pulse through darkness on bodies refined across more than 500 million years of evolution. The ocean itself remains what it always was, vast beyond practical comprehension, holding layer beneath layer of life inside conditions that erase shadow, absorb light, and reduce visibility until entire ecosystems pass unnoticed through the planet's largest habitat. For most of human history, the sea appeared empty beneath the surface because the things living there evolved specifically to leave as little evidence of themselves as possible. No silhouette, no reflection, no visible edge separating body from water. And yet the ocean was never empty. It was full long before there were names for the creatures moving through it. Full before the first instruments descended into mesopelagic darkness. Full before there were minds capable of wondering what invisible life might exist beneath the waves at all. That may be the quietest and strangest part of all this. The transparent animals do not become more real because they are finally observed. They were already there, drifting through cold darkness across spans of time older than forests, older than mammals, older than human thought itself. Tonight, after the lights above the ocean dim and the surface settles into darkness again, they will still be there, moving invisibly through the water, exactly as they always have.